Back, and today I'll be talking about the regulation of gene expression in prokaryotes. And microorganisms, they have the remarkable ability to adapt to the environment, and they can respond to the stimulus immediately by either turning on or turning off of the gene. And this is during the evolution, this process has helped them conserve energy. Otherwise, they would be unnecessarily making proteins which are not needed at that particular time. Now, regulation can be at different steps. As we know, it can be at the level of initiation, post-transcriptional modification, mRNA stability, translation initiation, post-translation modification, and so on. But for mostly, it is at the level of transcriptional initiation. Now, this regulation there are two ways by which a gene can be regulated. Either it can be turned on or turned off in a given environment, or they can have a cascade of gene expression or a gene hierarchy, in which if the first gene is turned on, it will then turn, if the first gene is turned on, it will turn on the next set of gene, a next set of gene, and so on. Now, one thing you should understand is when we are talking about gene regulation, we are referring we are not referring to the housekeeping genes. Housekeeping genes are those genes which are needed for the day-to-day maintenance of a cell. When we are talking about gene regulation, we are talking about those genes which respond to a particular environment or a particular stimulus. Now, E. coli can grow in a carbohydrate source of either glucose, sucrose, lactose, galactose, fructose, and so on. But in the first place, it will utilize only glucose. So when a glucose is present in the medium along with other carbohydrate sources, then all other processes which are required to metabolize those carbohydrates are turned off. Only the glucose is used, and once the glucose level goes down, and then the other sugar is used. Now, this process of turning on a gene expression is called as induction. And generally, induction of a gene takes place in a catabolic or a catabolizing pathway, where there is a substrate which has to be catabolized for use. And there's the other pathway, is the repressible pathway, which is generally used in an anabolic pathway, where a certain product is being made, but once its levels reaches a certain threshold, then it has to be repressed. So we have an inducible system in which there's a catabolic pathways involved, and repressible system in which the anabolic pathways are involved. Now, how these genes are turned on or turned off? These genes are turned on and off because there's another set of genes which are called as the regulatory genes. Now, the regulatory genes, what they do is they can bind to the regulatory elements on the operon, on the regulatory elements of the operon, and they can either turn them on or turn them off. Now, they bind to specific sites which are known as the regulatory protein binding site, RPBS, and the regulatory proteins, they can be activators or they can be repressors. So, activators, as the name suggests, they activate the gene expression, and repressors, they repress the gene expression. Now, for the activators and repressors to do their function, then there are effector molecules which are needed for their functioning. So, if a gene is turned on, then that effector molecule is called as an inducer. And if the gene is turned off, then that regulatory molecule is called as the co-repressor. That means it is binding along with the repressor to inhibit the system. Now, the Operon model was given in 1961 by Francois Jacob and Jacques Monod when they were studying utilization of lactose in bacterial cells, E. coli. And in the Operon system, what they have suggested is that all genes which are required in the same metabolic pathway, they are all regulated by one single regulatory structure, which means that the genes are stitched together and they are regulated by a common. So, if this is A, this is B, this is C, these three genes are needed in one pathway. They are all arranged one after another, which is regulated by the promoter region over here, the operator or the RPBS region over here, and all of them, they have a regulatory protein. Now, this regulatory protein is generally not involved in the operator. So, this structure is called as the operon. These are the structural genes and these are the regulatory genes. Now, to understand, if you what I'll tell you over here now is what are the two different types of systems that are we were talking about, the inducible system or the repressible system. Now, inducible system, as the name suggests, the genes have to be turned on, which means that the genes are turned off in the first place. So here the gene is turned off, and with the help of the regulatory protein and the inducer, the gene transcription is on. So in an inducible system, the transcription has to be turned on. On the other hand, the repressible system refers to that when the transcription is on, the transcription is generally on, and it has to be turned off. So when it has to be turned off, then it is called as a repressible system. Now again, you see I have written over here that in inducible system I have written negative control and positive control. What it means is, in the negative control, the regulatory protein is not needed for the transcription to be on. On the other hand, in the positive control, the regulatory protein is needed for the transcription to be turned on. Now, if you look over here, this regulatory protein, which is made by this regulatory gene, this is generally made in an active form. Okay, if it is made in an active form, it can bind to this regulatory region over here or the operator region over here. When this binds over here, the polymerase cannot sit over here in the promoter region, and the structural genes are not transcribed, so it is off. When the inducer comes, this inducer binds to this active promoter regulator over here, and it changes its conformation, which makes it off. One thing you should understand is that these regulatory proteins, they can undergo allosteric conformation. That means depending upon the binding of the ligand, they can change the conformation, which changes the ability to bind to the DNA, or whether they can bind or they may not bind. Now here this regulatory protein, so in a negative inducible system, the regulatory protein is active, which is binding, so there is no transcription, and the inducer changes the conformation. Now this cannot bind to the operator region. As a result, now polymerase is sitting over here and the transcription is on. On the other hand, in positive control, the regulatory protein is made in an inactive form. Okay. Now this inactive regulatory protein, it cannot bind to the operator. So when this regulatory protein cannot bind, the polymerase also cannot bind, because polymerase needs assistance of this regulatory protein. So there is no transcription. Now when the transcription is needed, an inducer molecule comes. Now this inducer molecule now makes this regulatory protein to be able to bind to this region. When this binds to this operator region over here, then the polymerase can come and bind, and the transcription is on. Okay. So what you should understand over here is that when we are talking about negative and we are talking about positive, we are talking about the regulatory protein. When we are talking negative inducible, that means this regulatory protein is not allowing the transcription to turn on. When we are talking about positive inducible, which means this regulatory protein is needed for the transcription to be on. In a repressible system, on the other hand, this transcription is on and it has to be turned off. Okay, so this is, they are repressing the gene expression. Now the regulatory protein in a negative control is not able to bind to the operator region over here. So as a result, now the transcription is on. Now when a co-repressor comes and binds with this regulatory protein, it changes its conformation. So when the conformation is changed, now this can come and bind to the operator region. So as a result, now polymerase cannot bind over here and transcription is off. In a positive repressible system, what happens? The regulatory protein, see, as when we talking about positive, the regulatory protein is required for the transcription. So here you see this is a regulatory protein which is needed for the transcription to take place along with the polymerase, and transcription is on. Here also you saw that this positive control, this protein is needed for the transcription to be on. So when we're talking about positive and negative, we are talking about the regulatory protein, their requirement in that particular process. Now here when the co-repressor comes and binds with it, this makes this structure, this protein can now not sit on the operator region over here, and the polymerase is not able to bind and the transcription is off. So here what we are talking about is the negative inducible and the positive inducible system. So lac operon, which is there in your syllabus, lac operon is an example of negative inducible, and tripoperon is an example of negative repressible. So in the next video, we'll discuss about these two operons one by one.